The next lecture will be by Dharm from my group at Ben-Gurion University. And Dharm's topics will be replication-driven peptide-based systems far from equilibrium.
Good afternoon, everyone. Today I'm going to present our research work in the title Replication-Driven Peptide-Based Systems Far from Equilibrium. This is the well-known question for everybody: how might life have been arisen? In this regard, O'Brien and Hayden given a hypothesis in which they thought a simple inorganic molecule, carbon dioxide, hydrogen, water, and ammonia, which react together and form the small building block molecules such as organic, bioorganic amino acid, nucleotide, and those molecules further reacted it give the polymer of the polymers such as the protein and nucleic acid. Those molecules assemble together and form the structure which were surviving, and it's form the and replicating, and it's form first life cell. However, they did not give any confirmation for their hypothesis. In this, then later in 1953, Miller and Urey did the experiment to test the hypothesis of O'Brien and Hayden. They have built a closed system where the which containing the heated water pool and mixture of gases. They thought that these gases present in the early stage of the atmosphere. They also have given the spark to maintain the atmosphere of the early stage. There, then they run the experiment for a week, and then they found there certain bioorganic molecules such as amino acid, glycine, alanine, and aspartic acid, sugars, and some other molecules found there in their experiment. And then in recent years, system chemistry has given great attention to the emergent life-like material. In this concern, organic molecules combine together by supramolecular assembly, and it forms the multi-component network which self-organize under the condition of out of equilibrium. It gives the emergent property, and which like prokaryotic cell, which is the simple biology. In the recent report from the Professor Hug group, they have designed a molecule which a flow-based oscillating network, where they did the combination of the time delayed negative feedback with the short positive feedback, and it gives the oscillating network. Then a simple reversible reaction which gives the in chemical equilibrium, it gives the single steady state. However, in bistable system, the rate of the backward and forward reaction, it is not similar, so that it gives the two steady state. To see, you can see here in this picture, you can clearly see here the A and V is the low steady state, and C and D is the high steady state. It is the a bifacial diagram of the bistability, and A and C, it is denoting bifurcation point. And this is the few examples of the bistability in nature. Bistability is widely used in digital electronic devices to store the binary data. And binary system can often switchable. They can flip from one state to other state by application of the specific stimuli. We have taken here two examples. One is the biological example, where a phage infected bacterium must choose between two cycles, a lytic and acyclic, to make the distinct lifestyle. This is the example of the chemistry where VZOAT systems, after light illumination, the phase image remains for about 1 minute, but the continuous lines transform into the dotted line. This is the one system of bistable. In this regard, we have designed a thiodepsi-based peptide non-enzymatic bistable system via replication process, where in each replication cycle template molecule or dimer where the electrophile and nucleophile assemble in its ligate and form the another copy of the replicator. Or which here you can clearly see the rate of the forward reaction and backward reaction means formation of the replicator or and decomposition, or it is not similar because of the formation of trimeric coil structure. It is stable, folding and stable. It's not decompose and give the electrophile and nucleophile, so that it's give the two steady states. One is the high steady state, another is the low steady state. To get the high steady state and low steady state, we have to design the experiment where we design here E plus R is equal to 100 micromolar and E equal to N, where we have clearly shown here, if we start the reaction from the low concentration of R electrophile nucleophile, it's give the low steady state only. If we start the reaction only with R or less amount of the electrophile and nucleophile, it's give the high steady states. And high steady state means high value of the K afferent. And then we can say from this figure, if we start the reaction from 0 or up to below 90, it's give the low steady state. In other case, 90 to 100, it's give the high steady state to make the system far from equilibrium. And you can see here, after the one cycle of the replication, the formation of the small molecule thiol and present nucleophile which oxidize and it give the unreactive molecule which stop the replication cycle. To prove this, we have done the experiment without TCP, where you can see after 24 hours, the system get the resting state. Then we, after 96 hours, we added the fuel, which is the TCP, which reduced this unreactive thiol molecules, and which start the replication cycle. It's give the considerable amount of this molecule replicator, and it's give the desired one of the steady state, low or high. To prove this hypothesis in more detail, we have did the reaction two conditions. One is the without fuel, another one is with the fuel. With the fuel means with TCP, we found the two distinct low and high steady state. However, in the without TCP, we found only linear formation of it depends on the starting concentration of the material. Then after checking the TCP is important for the reaction, then we check the effect of the parameter on the delta Kfran of the bistability. We check thiol concentration, total amount of peptide, guanidinium, and temperature and mutation of that peptide. We can see clearly here the difference this delta K afferent value, it is increases when we increase the total amount of the peptide. However, it decreases when we increase the amount increases the amount of guanidinium hydrocolloid, which is the denaturing agent, and also it decreases when temperature is increased up to 30 degrees C. And we can also see here when we did the mutation in this peptide molecule, it's also decreased the delta K afferent. Then when we did the CD, what is the mechanism behind that? We found here if the unfolded structure of this replicator molecule, it's give low steady state, and when it is, then it's moved to the bistable zone, means it is giving optimum formation of folded structure, which give the replicate, and it's give the two steady states. If we increase the concentration of that replicator, then it's give only single state due to the formation of two folded structure. To prove this hypothesis, we also done the theoretical calculation where we found the two points, and one is the this is 2 bifurcation point where blue star, one is 2 value of the A, low and high solutions. Then we thought, can we make this bistable system to another demonstrate these two state to different property? Yes, answer is yes. As we can see here, in living systems, cell signaling is a part of any communication process that govern basic activities of cells and coordinate multi-cell actions. There is 3 modes of cell signaling. First is cytoplasmic bridge, through cytoplasmic bridge, and second is secreted molecule, and third one is the plasma membrane bound molecules. In this regard, then we have combined here this bistable system with nanoparticles, where we can see this bistable system, high steady state or low steady state, which work as a output input signal for the NRG, which is the nano formation reaction growth process, which give the as a output in the form of morphology, shape, and aggregation. To check this effect, we did the experiment where we got the desired high steady state and low steady state, where we separately given the nanoparticle with the reducing agent. It's give the nano arching in the case of high steady state, whereas in the low steady case, it's from the nanoparticle hyper branch nanorods. In this profile, we can confirm by the TEM images. You can see clearly when after 2 days, it's from the nano arching in the case of high steady state, and whereas in the hyper branch nanorod in the case of low steady state after 2 days. To check the aggregation of the nanoparticle by using this bistable system, we did the experiment which give the separately high steady state and low steady state. In that case, then we seeded the gold nanoparticle blue seed, which give slow aggregation in the high steady state case and fast aggregation in the low steady state case, which one we monitored by the UV over the time. You can clearly see here in aggregation, it's high in low steady case compared to the high steady case. This one we also confirm by the TEM images. You can clearly see here, aggregation is high in the case of low steady state compared to high steady state case. Then we can conclude here, we demonstrated the bistable behavior by non-enzymatic self-replication, revealed again the potential role to replication for developing smart materials for use in synthetic cells. The magnitude of the bifurcation is decreases with the increase of temperature, thiol concentration, and guanidinium hydrochloride concentration, but it is increased with increasing the total material. We further suppose the quantitative control over the bistability function and bistability magnitude reflect the delta K afferent in our system would provide tool to design suitable material. We also explore here synthetic chemical memory by non-enzymatic bistable system. The two steady state can transmit different pattern of gold nanoparticles. Then I would like to thank Professor Gonan Ashkenazi to give me opportunity to work in his group and his research group. These are the highlighted person who contributed work in this project. And I also thank to Israel Science Foundation for giving funding to work us. And thank you for listening.
Thank you very much, Dharm.
Dharm,
yes.
Are there any ideas that you are developing with
yes.
respect to how bistability is relevant to life as we know it today?
Yes. Actually, as I have shown several 2-3 examples there, means bistable system can regulate that cell division, you can say, and it is also can correlate with means origin of life also, means how it is signaling happen and it's cell division or something, then it's from that living cell or molecules.
Thank you.
Okay, thank you. Thank you, Dharm. Thank you for a very nice lecture.